introduction. So before getting started, I'd just like to acknowledge my supervisor, Dr. Ravi Menon, my advisory committee, the members of the Menon Lab, as well as the CFMM staff who played a big role in this work, as well as my affiliations and sources of funding. So to start off with a quick agenda, we'll first be talking about some background information on functional MRI, then fiber photometry and awake imaging. Then we'll get into the methods, results, conclusions, and future work. So starting out with the background information. So functional magnetic resonance imaging is an application of MRI where we can basically infer brain activity from hemodynamics. So the mechanism basically works as follows. We have a brain cell, and it requires some baseline amount of oxygen to continue functioning at baseline levels. And it gets that oxygen from nearby blood vessels, and specifically the hemoglobin in red blood cells. So the red ovals represent red blood cells with hemoglobin saturated with oxygen. And then the blue ovals represent red blood cells after some oxygen has departed and gone to the cells. So at the baseline state, we have this situation. And as the red blood cells lose oxygen, the hemoglobin becomes more paramagnetic, meaning that in an externally applied magnetic field, it produces its own field. And if we use a sequence that can detect this, such as T2 star, then the signal drops as it becomes more paramagnetic. If we compare this to the situation where the brain cells are active, the oxygen demand increases and signals are sent to the nearby blood vessels, which result in the diameter increasing and an increase of blood flow coming towards that area. And that flushes out the paramagnetic hemoglobin and results in an increase in signal. And this signal is called the blood oxygen level dependent or BOLD signal. And if only it were so simple such that with an increase in brain cell activity, it results in a proportional increase in blood flow and then a proportional increase in BOLD signal. But unfortunately, it's not that simple. We have to remember that when we're dealing with the BOLD signal, we're not looking at the brain as we typically think of it, but rather we're looking at the vasculature that courses throughout the brain. And so even though we know that neurons have an effect on this vasculature, glia do as well, and same with pharmacological substances and other factors, including and especially pathology. These all affect the blood in their own way. And so that's really it makes it very difficult to interpret the BOLD signal, and that's the main motivation behind this work. So what many groups are doing now is they're including other techniques into their experiments to better analyze the BOLD signal and figure out the extent to which the BOLD signal can be decomposed into different components. And so for this work, we're combining fiber photometry with the BOLD fMRI. And this is a technique, it's an optical technique capable of detecting neurochemicals. So it basically works as follows. We inject a fluorescent neurochemical specific receptor virus into the mouse brain. And simultaneously, we also implant this optical cannula, which gives us optical access to that site of the brain. And then we excite that area with an optical fiber guiding a particular wavelength of light. And then those biosensors that were injected fluoresce, and we can determine if that targeted neurochemical is present or not. And so in our case, we'll be looking at calcium. And then one last aspect to this work is the awake mouse imaging. And Rabia had already talked about the benefits of this, so I won't have to go into too much detail. But of course, it all comes down to translational value and avoiding the suppressive effects of anesthesia and being able to perform more intricate tasks on our mice that we wouldn't be able to do if they were anesthetized. And this is, of course, more similar to our human experiments. But of course, it comes along with challenges as well. So we'll have to deal with the motion and stress in other ways. And so the objective of this study is to simultaneously record calcium and BOLD signal in awake mice. On to the methods. So dealing with the first of the two challenges of motion, we have decided to implement a head post style restraint. So here we show the different versions of the restraint that we have come up with. from version 1 all the way up until the versions that we're currently using. Version 2 was inspired by literature, and then version 3 onwards just have our changes that we made to better the well-being of the mice when they're in their home cages. It consists of attaching basically a head post nub permanently to the skull of the mouse. This is a 3D printed mouse skull to scale and just for reference. And then once the mouse has to be restrained, we attach one of these adapter bars to the head post using a screw, and then fit the mouse into the body restraint. So the head would fit, or the adapter bar would fit into these two vertical beams, and the mouse would be secured just in between, and the body would be held just behind. The MRI single loop coil is shown just above. This is the CAD design, and then the 3D printed model is shown just to the right. And then to address the second challenge of stress, we're implementing, as Ravi was saying, a 9-day acclimation protocol, starting with day 1, just 5 to 10 minutes and 0 decibels of MRI audio, and working our way slowly up to the duration of the actual scan, an hour and a half, and 100 decibels of scanner audio in the background. And this means that the mice have a bit more time to get used to this environment rather than being stressed if we were to just restrain them for the full duration with the MRI noise at the same time. Now on to fiber photometry. So we chose to inject JGCAMP7F, which is a calcium biosensor specific to neurons, and we injected it into the dorsal striatum. This shows the full striatum, and the dorsal striatum is, of course, just the dorsal region. We chose this region because it's a nice big region. It's easier to get the injection in the right location. And also it has some really interesting functions that I'll mention in a few slides. We also implanted the optical cannula into the same site as the virus injection, and that's shown over here. And this has the safety cap on top of the cannula and the head post just behind. It's all held together with dental cement. We then waited 6 weeks for viral expression and then validated the fluorescence prior to scanning. And then what's left is to combine the two techniques. So we investigated susceptibility due to the optical cannula, and then we recorded simultaneous calcium and bold signals while monitoring the mouse behavior using, as Ravi mentioned, the MRI compatible camera that we have. The functional parameters that we used are a 1.5 second TR, 12 millisecond TE, and 400 volumes total. So that brings us to 10 minutes per functional run. And our resolution is 300 by 300 by 500 microns. The anatomical parameters that we used are a 5.5 second TR, 30 millisecond TE, and a resolution of 150 by 150 by 500 microns. Now getting into the results. So to analyze the effectiveness of our restraint device, this is a representative motion analysis from one of the runs from one of our mice. We're showing the translation up top in microns for the right, left, posterior, anterior, and superior, inferior directions, and then the rotation on the bottom in degrees. Again, this is the voxel size for reference. And you can see that the translation is well under 1 voxel size, which is the generally accepted amount of motion that we allow for these runs. And so that just speaks to the effectiveness of this type of restraint. Next, we move on to the fluorescence validation for the fiber photometry. I've got a short video here just showing the calcium trace, which is seen on top, the green trace, and then the control trace, which is blue. I won't go into too much detail about what this is, but effectively, if you see signal here, then it means that it's a calcium independent signal, and probably due to the fiber moving or some other calcium independent factor. And just pay attention to the correlation between the mouse movement and the calcium trace. You'll notice that they are highly correlated. And so it's kind of neat to see behind the curtain what's going on inside the striatum at the same time as the mouse's behavior. It turns out that the striatum is heavily involved in motion, so it's a nice validation experiment to see this. And we can also note that there is very little fluctuations on the control trace, meaning that this is true calcium signal. Okay, moving on to the susceptibility analysis. So on top, I'm showing a T2 anatomical image, and then on the bottom is a functional T2 star functional run. The red arrows indicate the location of the optical cannula within the brain, and the yellow semicircle, or the circles, indicate the sensitive region where we record fibrophotometry. The cannula goes right to this area of the brain, and this is the area of interest for BOLD as well, because it's where we'll be able to compare the BOLD signal directly to our calcium trace. So what we really want to see here is if the susceptibility from the cannula extended beyond the tip of the fiber into this sensitive region. But fortunately for us, it looks as though it doesn't. There is some significant susceptibility around the fiber, as you can see from the T2 star image in both the coronal and sagittal views, but it really doesn't extend below. So that's really fortunate for our work. And then lastly, I wanted to show just the two techniques together. So the calcium trace is on top. This time we only have the one trace because it's been corrected with the control trace, and then the BOLD on the bottom. And I'll play this. You should be able to see some correlation between the mouse movement inside the scanner and the calcium trace as well. But of course, we wouldn't be able to see too much correlation on the BOLD trace because it is lagged compared to the calcium trace, and that hasn't been processed at this point, but that is in the future work. So in conclusion, awake mouse imaging was achieved with motion well under the acquired voxel size, and again, that's 300 by 300 microns in plane. And fibrophotometry was implemented to record calcium activity in the dorsal striatum, and calcium activity was observed to be correlated with the mouse movement. And fibrophotometry can be used to record neurochemical activity simultaneously with BOLD fMRI. Future work includes determining the correlation between BOLD and calcium signals using an appropriate hemodynamic response function. And I say appropriate underlined because we can't just use a canonical HRF, since that would assume that the calcium signal is synonymous to neural activity, which isn't exactly true. And then we will also perform the simultaneous recording with other neurochemicals to better characterize the neurochemical basis of the BOLD signal in different cell types. So that concludes my presentation. Thanks for listening, and I'd be happy to answer any questions.